So I'm going to be introducing Dr. Margaret Holik Romanovsky. Dr. Romanovsky is an assistant professor of health and nutrition sciences at Brooklyn College and a public health researcher dedicated to addressing health disparities in New York City underserved communities. Through community-based participatory research and epidemiologic studies, her work explores health inequities among populations of African descent with a particular focus on chronic diseases and cancer screening. She also leads the Immigrant Health Lab at Brooklyn College. So please join me in welcoming Dr. Romanovsky to the podium.
Good afternoon, everyone. It is my absolute pleasure to introduce our keynote speaker, Dr. Mary Beth Terry. Dr. Terry is professor of epidemiology and environmental sciences at Columbia University. She is also the executive director of the Silent Spring Institute, a nonprofit research institute committed to discovery and translation of the role of environmental chemicals on cancer risk. For over 2 decades, she has led molecular epidemiological studies focusing on the role of genomics, epigenomics, and other biomarkers modifying the effects of environmental exposures on cancer risk and cancer outcomes. Please join me in welcoming Dr. Terry.
Thank you so much, and thank you so much for the invitation. It's a thrill to be here, and I look forward to our discussions. I have 2 emails, 2 affiliations, so feel free to reach out and use either email to reach me. All right, I have no financial disclosures. So as many of you know, environmental causes across the board have affected many chronic diseases. And so I just, as any good epidemiologist would do, I just list here for your, just for your recall, again, the chronic diseases that we're talking about that are affected by environmental chemicals, air pollution, et cetera, include heart disease, cancers, pretty much everything here, including stroke. As we learned over the pandemic, there's a large role for many environmental chemicals in affecting your body's immune system and how it responds to infection, and also how your body responds to vaccines. And so there's a lot more that needs to be studied in terms of how these environmental chemicals affect treatment. And that's some of the work we're doing now at Silent Spring. But what I'll talk about today is more how does it affect risk of these outcomes, and specifically cancers, with a highlight on early onset cancers, which is an area that I do research in. So when we talk about environmental risk factors for chronic diseases, this morning's session on tobacco was a really fantastic one. Just to remind everyone that vaping products, in addition to nicotine, do increase your risk because of the heavy metal exposure. And the heavy metal in vaping products heats up, and that's a major source of carcinogens for people that are using vaping products. Of course, many of you are well familiar with pesticides and pesticides that can increase a whole host of different kinds of cancers. We also have VOCs, or volatile organic compounds, that can affect cancers and other chronic diseases. You have probably heard in the news, just last week, one of our chemists had an article that received a lot of news about the chemicals that are in hair braids. And again, these chemicals are part of a large class of what we call endocrine disrupting chemicals, or EDCs. And these chemicals can increase cancers both as direct carcinogens, or cancer causes, what I'll call carcinogens throughout this talk, but they also interrupt and affect hormonal signaling. So that's why they're called endocrine disrupting. I will mention, for those of you who aren't aware, but most chemicals, in fact, it's very hard for me to think of one that's not in this category. Chemicals are not tested for their effects on the mammary gland. So they're usually tested for acute toxic responses. And so most of the chemicals that we see in everyday products are not tested for their effects on the mammary gland. And then, of course, as you're well aware, and the accumulating data over the last few decades on how air pollution affects lung cancers, but now actually some of the other cancers that we didn't know air pollution caused, like breast cancer, there's finally enough data to support that as well. So the chemical class that's in the news a lot are these forever chemicals, or PFAS chemicals. There's hundreds of PFAS chemicals. And the reason they're called forever chemicals is they persist and they last for a very long time. Blood studies have supported that everybody has some level of PFAS in their blood. There's areas around our country, certainly, and around the world where there's much higher source exposure. Usually that has to do with being close to industry, close to military sites, things like that. There was a large cancer cluster coming out of Camp Lejeune in North Carolina. Also, a lot of exposure through the water, but you can also get exposure to PFAS through the type of dental floss you pick. So again, there's plenty of products that can be used to replace products with harmful chemicals. Not all hair braids have flame retardants in them or formaldehyde in them. So again, the point here is that there are some ways to substitute based on the knowledge that we have. So the kinds of cancers that are related to PFAS include kidney and testicular cancer, thyroid cancer, and now more and more people think breast cancer, and we're doing some work in that area. So as you know, with other kinds of exposure, social and built environment, that environmental chemical exposure is not evenly distributed. Here is just, it's a few years old now, but it's a map. You can get more of the latest data on the New York State Department of Health. But as you can see here, that again, there's a lot of environmental justice communities where the burden from environmental chemicals is much higher than in other communities. One website that I will point you to that obviously it's always changing, so please do look it up, but it's a very helpful website just to learn what's happening at the state level. Fortunately, there are right now in New York State a number of different bills that are being discussed. One is a plastic reduction bill. I would encourage you to look that up. Another one is a beauty justice bill. And hopefully, again, there's a lot of lobbying against those bills, but there's a lot of support for those bills as well. And they do need scientists. They need medical doctors and scientists to really provide evidence that can help in these debates. So what I want to then spend a few minutes on is this concept of early onset cancers. And you probably have heard in the news, there's more cancer. What does that mean? Is it the environment? So I have been looking at early onset breast cancer for about 25 years now, primarily starting looking at the genetics. But what we found very early was that there's a huge range of penetrance, meaning probability for cancer genes. And so we had an excellent lecture early this morning about cancer genetics. Everybody should know that there's not a single cancer gene that's 100% penetrant, meaning 100% probability that you will get cancer, meaning there's a lot of potential to modify people's risks. So we're going to talk about early onset cancers, but specifically, what do we know about environmental exposures and early onset cancers? And then leave you hopefully with some intervention examples and hopefully some potential for collaborations for people who might be interested in collaborating with us at Silent Spring. So here's just the big picture schema of where we are in this country with early onset cancers. And, you know, for better or for worse, we usually use an age cutoff to describe early onset as opposed to later onset cancers. Here, we're just using the age of 50. Sometimes we use 40, but What you can see here is that early onset cancers are going up, but also that the predominance of them, in fact, 2/3 of early onset cancers occur in women, and that's primarily driven by breast cancer and then thyroid cancer. Colorectal cancer, what you've heard a lot about in the news because it is the #1 cancer mortality under 50. It's increasing in both men and women, and you can see that this has been going on for several decades. I don't have it here, but there's been a lot of analysis, including from our group, looking at specific birth cohort trends. And I'll cut to the chase and tell you that for colorectal cancer, the more recent birth cohorts, so kids that were born, I guess they're adults now, but from the late 80s forward have had a lot more cancer than earlier cohorts. And so this has led to a lot of people saying, well, is this real or is it just we're doing more screening? So one thing I want to just emphasize is, yes, there are more CT scans that are done, so there can be more cancers that are incidental, but these are ages where we're not doing any population-based screening. So some, but a very small part of this increase in early onset cancer is from incidental findings from using more medical imaging. What we did, and is published in October, was use the cancer registry data that have gone back several decades to specifically show, yes, the tumors are getting smaller, so usually that is from having better ways to detect cancer. But what we call the sojourn time, or from when you have the first cell that's affected by a carcinogen to when you get clonal expansion, that time has shortened in recent birth cohorts. And we found that to be true for breast cancer, we found it to be true for colorectal cancer, and we found it to be true for thyroid cancer. So not only are they rising in general, but the time between when you have the first hit, so to speak, and that clonal expansion is getting much shorter in younger generations than ever before. And so what is going on? So if we look at, this is just a standard schematic of what we know to be true for cancer, which is most cancer occurs when people are older. However, the incline or the steepness or the rate of change is occurring much earlier, and then it plateaus out. We do know most cancers take several decades from that first cell being initiated to when the cancer occurs, but that's on average. And like I said, some of these times, at least based on our models, are shortening. And so cancer has been and is described as a disease of aging, meaning this green bar here is showing, as we move forward in life, we know that aging is not a linear process, that, eventually we all die at different, but this rate is pretty much in general mirrors the overall population cancer. So that is why people say that it's a disease of aging. And if we see more cancers, believe it or not, I've actually had people say to me, well, we see more breast cancer, that's a good thing, isn't it? Because that means people are living longer. And that is quite the opposite of a good thing, because again, when people are getting cancer, especially young generations, they're getting more aggressive cancers. And so it is true that if you live long enough, like, autopsy studies going up until about 100, most people will die of cancer, not die with cancer, not of cancer, not from cancer, if you live until your late 90s. So everyone has some sort of cancer later on, but those aren't the cancers that are the most aggressive. The most aggressive cancers are the ones that are diagnosed much earlier. And the other thing we've learned over the last 20 years, these brown dots here mimic what we call somatic mutations or acquired mutations. And so when you talk to a molecular biologist, they'll say all cancer's genetic. And that seems like an anathema to people in public health, because we like to think that we can modify and prevent or have risk reduction programs to reduce the risk of cancer. And in fact, we have great examples of that with smoking cessation and lower lung cancer rates. And Also thinking about vaccination and lower cancer rates. So not all cancers are going up in young people. Cervical cancer, fortunately, is going down because of the human papillomavirus vaccine. But what are these brown dots? So these brown dots are actually an estimate of the somatic mutations. So when molecular biologists say that all cancer is genetic, what they mean is changes in the genes. But most cancer, in fact, anywhere from 90 or even 95% of cancers are from somatic or acquired mutations, not germline mutations. And again, that will differ based on the cancer type. So, but what's interesting, I think, here from this paper, and it's already now 10 years old, but what they estimated is that half of these somatic mutations that cause cancers occur before your body's fully mature. So usually people estimate that to be like 25 because the brain is actively changing until you're about 25, which is why I always told my kids, don't start drinking until after 25. But, you know, it's hard to tell kids sometimes things, but anyway. So, you know, so what you can see is a lot of these somatic mutations are occurring. They're not following this pattern you would expect. So that both tells us we need to do cancer prevention earlier, and it also begs the question, what's driving these somatic mutations? So what we think is happening in younger birth cohorts, so birth cohorts, we looked at birth cohorts of people born in the 50s versus the 60s versus the 80s. What we think is happening is this accelerated cancer because of accelerated aging processes. So if you will, if cancer is a disease of aging, these younger cohorts are aging more rapidly. And that's driving the somatic mutations. And so it's a concept of biological aging as opposed to chronological aging. And so related to that concept of accelerated aging is this idea that the other thing that's changing much more rapidly or differently rather, is in the younger cohorts is the immune environment. So again, differences in childhood infections that people are exposed to, differences in the use of antimicrobial products, differences in the gut microbiome, differences in the chemical environment, which can affect the immune system, as I mentioned already. So over the last couple decades, if you're old enough, you can say that about your research program. And the last two conferences I went to, I was tasked with just giving the history talk, which can be fun sometimes. But anyway, the history of the environment in breast cancer research, if you will, supports that actually it's a pretty consistent story with environmental chemicals in breast cancer. A lot of people have described it as an inconsistent literature, and I would say a large part that's for two major reasons. One, because most studies were not conducted when the breast was changing in form and function, and that is what we call these windows of susceptibility, or WOS. And so you can we're exposed to carcinogens all the time, and you can get cancer any day of your life. I am a buzzkill at most parties if I talk about this stuff, but there are more susceptible windows. So even though you can get cancer any day, you can be exposed to a carcinogen. In fact, we're exposed to hundreds of carcinogens every day. Depending on the cancer type, there's times in your life where you're much more susceptible. And for the breast, it happens to be when you're young. Babies go through what they call an infant puberty. So all babies, regardless of biological sex, grow breast tissue, and then the breast tissue regresses. So whenever breast cells are growing or contracting, changing during puberty or pregnancy or menopausal transition, those are when your breast cells are more susceptible. And when you look at those windows, incredibly consistent literature. The second reason the literature is much more consistent than just simply counting up the papers that have looked at environmental chemicals in breast cancer is when you do look at And here's just a variety of different types of chemical compounds we looked at, considering legacy pesticides and air pollution, personal care products, that the literature is much more consistent when you factor in somebody's underlying susceptibility. So for the most part, that would be based on what we can measure more easily, which are germline genetics, family history, whether or not someone has an early onset cancer or not. But in these populations, contrast to what was the paradigm 30 years ago, which is if you were diagnosed with a young early onset cancer, people thought it's just your genes, and if you were diagnosed later in life, it was just the environment. All cancers are an interaction between genes and environment, all of them. So what we found, though, is in these young individuals that the consistency was much more consistent when you looked at people who are more likely to be susceptible. Now, that doesn't mean that it's not important for later onset cancer. It just means that the signal of what we can detect is much more consistent. We did this in several different cohorts. This is an example of a measurement from the blood looking at polycyclic aromatic hydrocarbons, which we're exposed to through air pollution and sometimes through the cooking method when we barbecue our food and things like that. That if you had high exposure to these compounds measured in the blood, and you also on the x-axis here is describing the absolute risk of cancer, you were much, much more likely to get breast cancer. So again, in these areas where the odds ratios are 2, 3, 4, 5, it's very unlikely that these can be explained away by chance or some other kind of bias. We've replicated some of this now in a much larger cohort, and you can see in this young women, it's much easier to detect a signal. The signals still exist, for older women, but you can see when you're in when you have people where most cancer is diagnosed, which is later in life, you also have a much greater heterogeneity when it comes to absolute risk. So again, we kind of consider these early onset cancers as not only informing what's driving cancer for these young cohorts, but also helping us understand later onset cancer, where outside of an occupational setting, it's much harder to detect the chemical effect in people who are exposed to hundreds of chemicals a day. And it's hard to measure them as well. This is, in the interest of time, I'll probably skip this, but this is some recent analysis. I'll skip the detail, but this is some recent analysis with DDT, which was a type of pesticide that was banned in 1973, but is still being detected in dust of people's house. So that just shows you how persistent some of these chemicals can be. It also is one of our top 10 tips at Silent Spring is to dust, So I know when I grew up, there was always that saying, the cleaning and scrubbing can wait till tomorrow. For babies grow up, we've learned to our morrow. So it was basically saying, don't worry about dusting. But you should worry about dusting because there's a lot of chemicals that can be stored in dust. So anyway, but this also shows that it's pretty complicated, some of these gene and environment interactions. But most of these organochlorine pesticides were very much related to increased breast cancer, and some of these were decreased. So why then is this important for later onset cancer? So back to this kind of schematic of how we study cancer. This again is just the standard kind of curve for any cancer. What's represented in red are what I call again these acquired mutations, or specifically somatic mutations. And what's in green are these germline mutations. So germline meaning you inherit them from your mother and your father. And what you can see is when we do studies of people where most cancer occurs, we're getting a lot more contribution from somatic mutations, which is okay if you can actually measure them, but it's much more difficult to measure somatic mutation. You actually need the tissue. So for breast cancer, you'd need the breast tissue, and you'd need to measure the somatic mutations in that breast tissue, as opposed to when we're measuring germline mutations. You probably know if you've done any genetic testing yourself or had any done for yourself, you can just spit in a tube and get your genetic testing. So, but what you can see is the way the cancer epidemiologists have done a lot of their research is by getting people mostly here, and they miss all the people here who are more susceptible, had more environmental exposures, and maybe had a comorbidity and then didn't get, or another cancer, and then didn't get included in the study. So we miss a lot of the people that have the most susceptibility as well as the higher exposure. So I do think that's a problem with how we've done a lot of cancer epidemiology studies in the past. So stick with me, if you will, for just this one slide, because this is a paper that a group published during the pandemic, and it's really kind of changed my own thinking of how we do cancer studies, because sometimes a picture is worth 1,000 words. So on the left-hand side, what do we see? So in pink, this is what the last 25 years of the Human Genome Study and all of the cancer studies have taught us, which is there's over 1,200 genes for cancer. Those are represented in pink. There's about another 300 more that are somatically mutated. So mutations that you can't inherit from your mother or your father. But there's a huge overlap, except for 4 genes, which these are, I think, genes that contribute to lethality very early in life. But all genes that are cancer genes that you can inherit from your parent, those same genes are the ones that get changed somatically throughout life. And so that's important because, again, most people are going to have most people have cancer from acquired mutations, not from germline mutations. And then what happens as you go forward, and as you age, the proportion of the germline mutations, the proportion goes down as you age. So as you age, you acquire more family history of cancer, but that family history is less relevant to predicting if you'll have cancer. So because as you age, the proportion of cancers that are explained by germline go down. And the exact opposite pattern for when you age, the proportion that are explained by somatic mutations go up. So the other thing that I think is really important to communicate is that somatic mutations or acquired mutations, those can be driven by 2 things. They can be driven by pure random, sometimes people call it bad luck, right? Every time a cell divides, you have a possibility for a mistake. And so about 10 years ago, there was this huge debate in the literature between people who said all cancer is bad luck, and then the other people said, no, all cancer is environmental. And the reality, it's both, right? But what is important for us to think about is when we see trends of cancer going up in young generations, that can't be explained by bad luck, because the bad luck probability was there in the 50s and the 60s and the 70s and the 80s. So the only thing that when you see trends, it can't be by germline changes, because that they don't change that often. Germline genetic changes go over hundreds of years as people, people marry and populate and all of that, but not over just a few decades. So it has to be environmental. So there's always, somatic changes will always be both, but we need to stop arguing about is it that or this, because it's both. But we do know, and we have to really communicate that effectively, that the increase in early onset cancers in the US and actually globally is not because of bad luck. It's because of the environment, and we need to know then how to stop it. Okay, so I do want to then end the last 10 minutes with what can we do about it? Because I think it's important that, we think about knowing now what we can do now without knowing all the information. Just like before we knew everything, every kind of carcinogen that was in cigarette smoke, we could still tell people to stop smoking. So I want to contrast this approach, again, with it's not one way or the other, but it's both. So what I mean by that is in public health, we often take this population-based approach, right? Vaccination, for example, has to be population-based or you don't get herd immunity. The contrast to that is what we call a targeted approach. And so a targeted approach is about finding the people at this point of the distribution that have the most risk and doing something about it. Population is saying, I want to move the whole population one way or another. And so that's about changing the average. And so what I think is helpful to think about is just like we think about, and this is the cancer control continuum, just like we think about for precision medicine and tertiary prevention, that there has to be both, right? So we have to, on a population level, have better access to care, support interventions on physical activity or diet or whatever survivorship program there's going to be for everybody. But of course, we know that for some individuals, based on their somatic mutations or based on their risk group, that there are very specific targeted therapies. So it's not either or, but both. Similarly, we've changed our thinking on screening. So even though population-based screening has been incredibly effective for certain cancers, primarily breast and colon, for screening, pap smears too, based purely on age, more and more people are advocating for risk-based screening, even for mammography to go to a plan based on your 5-year risk of breast cancer. So again, there's arguments to have to use both. So I would say as prevention scientists or people who do risk reduction, it's the same sort of thing, and we need to be open to thinking about it at that level. So for example, what do we want to do for our population as a whole? We want to do vaccines, HPV vaccination, hepatitis B vaccination. We want to have regulations against tobacco. We want to have regulations against PFAS, which we have had, but some of them are being pulled back on. We want to have regulations about clean air, which we have had, but some are being pulled back on. And again, other kinds of preventions that you can recommend for everybody. But then there has to be also a role for targeted interventions, including environmental exposure reduction. And so even though I'm going to now just talk about some of these interventions, I want to be very clear that the best way, just like with smoking, is through regulation. That's how you get the most bang for your buck, if you will. But it doesn't mean that we can't do something now with a hybrid approach. So what I want to talk about then is what I call cascade prevention. And it's coined from this concept of what we do in genetics, which is cascade genetic testing. So when we do cascade genetic testing, it means if you have a relative with cancer and they have a germline mutation, and usually it's based on testing for people who have early onset cancers. So for example, for breast cancer, if you're diagnosed under 45, you will have certain tests. Now that age has gone up as the cost of genetic screening has gone down. So there's some cancers now that you'll test everybody, and that also has some treatment implications as well. But then based on that test, doctors will go and find all their first-degree relatives and then give them a test. So that's cascade genetic testing. It's happening now. So again, it's done, and the NCI has the latest information on each cancer by itself. So cascade prevention is the same idea in my mind, which is just like we do with genes. If someone comes to the clinic with an early onset cancer, we're finding the relatives anyway, to test their genes, their germline genetics. We could do prevention interventions then as well. And again, it's not either or, but at the very least, it can augment population-based approaches. So, and again, just for emphasis, that the family itself is more than just the genes. The family is also the environment. And if you can change something, we do this all the time, thinking about certain certain guidelines for women when they're pregnant. If you change something during this window of susceptibility for the mom, it can both prevent chronic disease and specific cancers for her during that window and after that, as well as for then the baby. So again, thinking about ways to intervene through this network, which is genes, but then so much more than genes. What we've also learned is just how important not just the influence of your parents, but also your grandparents. And that's a lot of the work we've learned through epigenetics. And so this intergenerational transmission, if you will, of the environment. So sometimes people will say, oh, well, I don't understand why you say air pollution is related to breast cancer because we have the cleanest air we've ever had, and that makes no sense. And one of the things I usually say back is, well, first, breast cancer takes a long time, but also it's thinking about the exposures the person has, but plus their mother, plus their grandmother. So again, thinking about these networks. And then also just really emphasizing the difference of our approaches to methodology for finding the causes versus prevention itself. So when we think about intergenerational health, again, we have been doing interventions during the pregnancy window for the mom, and then following up the mom as well as the baby for changes in breast tissue over time. We've done that in a few different studies. And what I would say is that it's important just in general for prevention scientists to think of this interconnectedness between how the family's influence and the social influence and how that actually changes across the life course. It's never constant. And of course, families themselves have more influence with thinking about vaccines, early life exposures, thinking about nutrition, macro level factors. Social networks themselves are very influential, especially during the adolescent time window when addictive behaviors are starting. And so I'll leave you with one finding that we have been working on recently, because it connects to some of these personal care products, is we've been following girls and their moms and the girls, I should do it in that order, from the early 2000s. And so this is a mom who was wearing a backpack to catch ambient exposures in her indoor air environment, and she wore it for 24 hours during her pregnancy. And then we follow up both the mom and the daughter to look at their breast tissue changes 15 years later. And we actually did find, first we found in utero exposure to air pollution and polycyclic aromatic hydrocarbons predicted the daughter's dense tissue during adolescence, as well as increased collagen levels, which deals with the elasticity of the breast tissue, which is a risk factor for breast cancer too. And these are some of the chemicals we were finding. So this data is not published yet, but hopefully will be this spring. But again, supporting the fact that you can have this window of susceptibility that can be very influential later on. So what can we do about it? So there are a lot of household interventions. I'm going to just, and I'll share my slides if I can, I'll figure out how to share them. The file got kind of big, so it was hard to share. But in collaboration with We Act for Environmental Justice, we've done some household environmental interventions. We're hopefully doing some more now with NYCHA. But we have at Silent Spring what we call Digital Exposure Report Back Interface, or DERBI. And we have now a way of translating this information and prioritizing it, because it is a few, you can get easily overwhelmed by the number of chemicals we're exposed to, but the way these digital tools work is they prioritize which ones are the most important for you and your apartment. And again, you can make changes pretty quickly with a few steps. One of them is probably one that many of you already do, which is never wear shoes in the household, because shoes track in a ton of chemicals from the street. Another thing which I'll never abide by because I love my dogs so much, but pets can get a lot of the chemicals in their fur, and you can just get a lot of exposure yourself that way. But pets are healthier for getting you out of the house than walking, so you gotta you gotta weigh your stuff. So anyway, I'll share these with you so that we have some time for questions. But we have also web-based tools for doctors and community members. We have a web-based tool called PFAS Exchange. These are all free, and I can share the website with the organizers. We have a Healthy Home logger that you can log your tools. We have had for the last 8 years, we're revising it now, but you can still download the current version. The new and improved one should be ready in a couple months, but it's called Detox Me. It's free. You can get it in the App Store. Again, it'll give you some ideas of things you can do, particularly to lower exposures for kids, to change, think about clothing. A lot of clothing does have flame retardants in it, especially for young kids. And again, that was started for a good purpose, right, to protect the child, but the amount of chemicals that are in them is not necessary at that level for protecting. So again, things to be thinking about that are immediately within your own control. And again, for those people who may not use a lot of personal care products, but still want to go through, your apartment and think about ways to reduce things. Right now, New York State has a law on the books that your doctor can prescribe you an air filter. So if you have a chronic disease, your doctor can write you a prescription, and that can be free, and you can put that in your bedroom. And if you don't have a chronic disease and your doctor can't prescribe one, you can make an air filter, and there's some online tools that are free. It's usually definitely less than $100 to make an air filter, and that can improve your indoor air environment. And then finally, we have this Detox Me logger, which you can put in your products, and you can find out a little bit more, because we firmly believe that you shouldn't have to have your PhD in chemistry to be able to know what's on your product. And so these tools are there to help you, and they're free and they're online, and I'm happy to share any of those with you. And again, I would encourage you, if you're really interested in PFAS, this particular website just was revised and relaunched this past fall. And I share it with all of my clinical colleagues because it's been very helpful for them. It also, if you're in a PFAS, a heavy PFAS exposure area, it will tell you how to get your blood tested. And I will then leave you with that, and love to keep the conversation going. We have a multi-institutional center if you want to work with us on any of that, as well as at Silent Spring, and leave you with this idea that there are things we can be doing now, both within our own personal environment, but also at the state level. So if you need more information there, I'd love to talk to you. So thank you, and thank my collaborators. Okay.
This was amazing. Thank you so much. I'm going to turn my air filter back on. It was on when we had smoke conditions from Canada. Thank you, Dr. Terry, for this amazing talk, very inspiring, and I took pictures of all the tools. Now we're opening up for questions from the audience.
Yes.
We have a question up here. Yes, ma'am?
Hi. Is there any evidence that you may have about the possible impact of the COVID virus in this whole discussion?
So it's not my research, but my colleagues' research at Silent Spring was looking at how PFAS chemical exposure limited the effectiveness of the COVID vaccine. So again, a lot, and it's not just her work. I've seen a lot of papers look at things like that. But yeah, so many of these chemicals, it's a great question. Many of these chemicals don't just affect your cancer risk, but do affect like your immune response, which can then affect your cancer risk. So yeah, that's a great question, yeah.
Thank you. I realize I'm in the light here. It's
Yeah,
really
it is
hard
hard.
to see.
So,
There's a question over here. Yes,
yeah.
please.
Good afternoon. Thank you for an excellent talk. So the TCGA results that you saw were striking with the opposite trends between the germline and somatic mutation plots. So I was wondering, so genetics research suggests that not everyone acquires somatic mutations in the same manner, but rather the people that they have higher number of germline mutations, they tend to get more somatic mutations over the course of their life. So I was wondering if you think that correcting for background germline mutations actually would have a different pattern in the somatic mutation plot, or how can we integrate this kind of knowledge in trying to predict how many somatic mutations someone acquires or how this might interact with the environment?
Yeah, I think
general.
it's a great question. Yeah, I don't, other than resources like the TCGA or the state of Maine, I think is one of the first states to have population-based data on somatic mutations. So we're working with them now just to look at whether or not there's a difference in the type of somatic mutations based on rural versus urban exposures. But yeah, until recently, the TCGA is the only place that I know would test something like that. And I don't know that the data are, it's a great question. I just don't know if the data are linked that you can look at that longitudinally like that. Maybe a patient population that's well annotated, but yeah, it's a great question. Yeah.
Thank you. We have a question in the back row.
Hi, thank you for the insightful talk. So I had a question about your research about the mother and daughter pairs that you showed, and in response to the exposure of the mothers, the daughters seem to have like denser breast tissue characterized by like increasing collagen and stuff. So I wanted to know your insights or thoughts on is this like more of an epigenetic change? Is this in response to like an inflammation response that's like in response to the, I guess, pollutants or whatever is the mother is being exposed to?
Yeah, it's a great question. We don't have, we don't have an analysis of the specific epigenetic changes in this population. It is a small study. It's about 200 moms and daughters, but what makes it kind of unique is that it's not based on any report or anything. It's measurements of the breast tissue and measurements of chemicals from either from a backpack exposure, like it's all objective kind of measurements. So what we did find, which I didn't explain because of time, but I do think it's interesting, is that, so we did a full, now the state of the art is such that we can measure hundreds and hundreds of chemicals at the same time, which is what we did. So we did an exposomic analysis of the mother's breast tissue and the daughter's breast tissue, and there was a lot more hits with the daughter, because the moms all are moms, so they've already gone through a pregnancy, at least one, so they've had their breast cells differentiated. So whereas the daughters were teenagers, and so there was a lot more hits, so to speak, and the chemicals were explaining the variations. You know, I think one of the analyses, it was like 65% of the variation in the daughter's breast tissue were explained by these environmental chemicals, and not based on, like people like to always say things are, you know, attributed to the things that are easiest to measure, like smoking or body mass index. But these kids, it was not related to body mass index or smoking or anything. It was related to these chemicals. Yeah.
Hello.
Well, right.
Hey.
Yeah, go ahead.
So, my name is Hara Prasad. I have a question. Actually, I have two questions. One is silly, and the one is science. So the first question.
Science can be silly.
So my first question is like, how safe is the drinking water or the tap water of USA?
New York City, it's some of the best in the world. We're lucky. Yeah.
Oh, that's good then.
No, we are.
Okay.
It
I
is
was knowing
true. We are very lucky here, yes.
Yeah.
It's not true on Long Island or, again, there's so much variability, and it depends on if you have city water or you have well water. And a lot of parts of the country, they have well water, which then you should really be testing your well water, because that's how you can get exposed to a lot of chemicals, because they stay in the soil for a while. Yeah. But we have very clean water. In fact, I just because one of my kids was interested, and some of these like water testing kits you buy online are not not terribly comprehensive. But you can buy a $10 water testing kit online, and at least where I live in Washington Heights area, my water was better than the water in these plastic bottles we have here.
Oh, really?
My water from the tap was better.
But so I use tap water. Yeah.
So we can go and directly drink tap water, no problem.
Well, again, like I think if you're worried, test it. But New York City water is some of the best in the world.
Okay, the question of science is like, is genetic engineering like CRISPR-Cas and all is allowed to be done on like humans, or because we are seeing that a lot of genetic mutations and all. So if we get the disease early, can we mutate and like edit the gene and then create a new thing, like a new life, give a new life to completely eliminate the cancer-causing gene? I don't know.
Yeah, I mean, I know obviously the gene editing with muscular dystrophy and with sickle cell have been pretty incredible breakthroughs. I think cancer is way more complex, so I don't know. Yeah, but I don't want to speculate. Yeah.
All right, we have a question over here.
Hi, Dr. Mary Beth Taylor. I'm Nyasha Chambwe from Northwell Health. So I have two questions. The first is, it seems technology has developed to the point where you can get more specific individual measures of exposures. And so my question is, if you don't have those measures, how well do those measures correlate with area level measures that we've been using in population
Oh, that's a great question. So data science, as you know, and I love your work, yeah. So we're at a place where we're at such an incredible time, I think, right now for understanding environmental exposures and cancer generally. We being the field had occupational studies, that showed asbestos and vinyl chloride and, all of these other kinds of occupational exposures. But what's been super difficult until now has been to measure environmental chemicals that we're exposed to every day in a non-occupational way, like through the products that are not regulated, through the air that we breathe, the water we drink, not in New York City, but yeah, in general, right? So there's two things. I think the area level stuff is crucial, and that can be done in a very big way and can be done for policy purposes, which, and just like the past couple months as a reviewer, I've seen really great area level stuff where like dose response distant to a nuclear power plant, obviously proximity to Superfund sites and these sorts of things. And the studies, at least I remember, accepting, and I guess they'll be published soon, looked at not just do people in like proximity to Superfund sites and to nuclear power plants and other sorts of environmental exposures, do they have more cancer, but they looked at do they have more metastatic disease. And they are more aggressive cancers, too, with environmental exposures, which is what we're trying to do at Silent Spring now, is look more at how they interfere. Our hypothesis is that they would interfere with immunotherapies because they affect the immune system, or they would interfere with endocrine treatment for breast cancer because they're endocrine disrupting. But those are all hypotheses right now. But I do know there's a group in Michigan that published a really interesting paper a year ago, I think now, where if you expose cells to endocrine disrupting chemicals, it changes a breast cell that's a luminal cell, which are, the hormonal responsive cells, to a basal cell, which is a hormone non-responsive cell. So if that is true, then in human studies, then that would suggest that there's a potential, like through reducing these endocrine disrupting chemicals, to lower the burden of basal cell breast cancer. if that makes sense.
Yeah, that makes sense. I think we're part of a mutual admiration society here. So my next question is about the gene environment type studies. So you said, okay, we like some of the data you showed, you stratify or account for baseline germline risk, and then the environmental exposure is layered on top.
Yeah.
So I was wondering, I always think of the germline component as germline risk for cancer, but have people asked the question whether it's germline related dysfunction is in processing or detoxifying those exposures as well?
Oh, yeah, no, that's a very good point. I mean, if you have, if you already have one gene, like a BRCA1 or 2, that lowers your DNA repair capacity, right? Then you would expect, yes, then that's maybe increasing then your inability to repair the damage, from everyday exposures. Someone at a talk had said everyone has a cancer cell in their body every 3 days. It's just we repair it, get rid of it, blah, blah, blah. But then after she gave that talk, I said, give me that citation. And then she could never find it. So I don't know if it's actually true, but it is interesting because, why aren't we all walking around with cancer? It's because we do have good DNA repair capacity most of the time, right? And yeah, but that's a good question. Probably they are interacting, you would expect, yeah.
Thank you so much, everyone. Let's give Dr.
Yeah.
Terry another round of
Thank you.
applause. And thank you for all the great questions.
This is the last session, and we're going to have the flash science session. We're going to hear from some wonderful people who will tell us about their science and what they are doing to solve the problem of cancer and maybe cancer health disparities. And to moderate the session would be Dr. Prem Surut. She's a physician scientist and associate professor in the Department of Cell Biology at SUNY Downstate Health Sciences University. Her research bridges molecular genetics and translational oncology, focusing on tumor suppressor pathways, cancer resistance mechanisms, and immunomodulatory therapeutic targets. Her work integrates molecular biology, cancer genetics, and immunology to advance innovative diagnostics and therapeutic strategies in oncology. So please join me in welcoming Dr. Prem Surut. She will introduce the speaker.
Thank you. All right, so it's always nice to be last up. So we have 3 speakers. I'm first going to welcome Dr. Cassidy Lundy-Perez. Dr. Lundy-Perez is a computational biologist at the Feinstein Institutes for Medical Research at Northwell Health. Her research integrates genomic and single-cell approaches to better understand endometrial cancer and other gynecological diseases across diverse populations. So again, her talk today will focus on her work and risk models in endometrial cancer. So we'll keep these talks to about 12 minutes or so, 30 minutes, and then open it up for questions after each talk. Thank you.
Thank you so much for the introduction. So today I will be talking to you guys about my work in the Chomboy lab studying endometrial cancer. So in recent years, many cancers have seen a decline in new cases and in mortality per year. And subtypes such as lung cancer, liver cancer, and prostate cancer show a continued decline in new diagnoses and in mortality. However, this overall trend of decline falls through in the case of endometrial cancer, or EC, where we actually observe the opposite trend, where new cases and mortality are still unfortunately on the rise. And furthermore, high-grade endometrial cancer is stratified by race. So when we look at this source here, we see consistently higher cases of the proportion of aggressive endometrial cancer within the non-Hispanic Black population. And so while these statistics support a need for race and ancestry-aware endometrial cancer studies, current studies do not always account for ancestry and race in existing risk models. And so here is kind of a scope of some of the existing risk models where we see that there's a whole host of covariates that are accounted for. And so they're kind of stratified here on the right. And even more, like grouping them even further, we see that there are some clinical variables, there are some genetic variables, and there are some that can be classified as social. And so we hypothesize in the Chambwe lab that the germline genetic variation and the social determinants of health are significant predictors of endometrial cancer risk across populations. And so for that reason, we're particularly interested in variables such as race and genetic variation explained by, for example, polygenic risk scores, or germline variation explained by other analyses. And so if we look for the intersection between all of these different components, like any two of them or any three, there actually is no one model that incorporates all of these different covariates. And so we believe that our proposed model will provide unpublished insights to the field. And so in our data, we primarily are going to focus on the genetic variation components in the coming slides, but we'll also kind of mention a little bit of our interest in the social determinants of health metrics. So in order to test our hypothesis, we wanted to build a custom and diverse cohort from public available data. And for this application, we used the All of Us research program. And more on the All of Us research program, it's particularly interesting for the fact that it has relatively high proportion of non-European or more diverse populations. And it is also more of the one of the more recent cohorts to be published. Additionally, within the All of Us cohort, we see the inclusion of both genomic and endometrial data, which is particularly useful when building integrated models. And so EHR data, or electronic health records, as well as survey data, was used by Claire, who is here in the audience from our lab, in order to build a novel algorithm for generating a more diverse cohort with the All of Us consortium. And so this schematic here kind of shows some of the flow that she used, which essentially leveraged EHR data in order to curate a cohort that was robustly associated with endometrial cancer. And this cohort is what we used for the foundation of our genetic analyses within our diverse cohort, such as polygenic risk scores. So just really briefly, as we know, there are many common traits that are influenced by thousands of different genetic variants. So in some cases, we have diseases that are only really caused by one particular variation at one particular loci, but more common are these more complex traits. And so a polygenic risk score, or PRS, combines the different small effects of all of these different variations across the genome, and it summarizes these different effects into a single number that kind of will signify the risk that a particular individual has to developing the disease. And so these scores can actually be heavily influenced by the genetic structure of the input data. And there are a couple of different steps within the PRS analysis where we can account for the effects of the ancestry structure of the input data. So for the next few slides, I will be talking more about how our initial PRS analysis and how we performed this analysis in a comparable All of Us diverse cancer cohort. And then I might refer to the test cohort as published variants and refer to the validation cohort as just our all of us cohort. So one of the initial questions that we had is, does polygenic risk scores, or do polygenic risk scores generated in a homogeneous population adequately model risk in a diverse cohort? And so one of the first things we wanted to assess in our results is, does the polygenic risk score distribution follow a general expectation? And so this is distribution of our polygenic risk scores when we scored our cohort. And we saw that it was normally distributed, and we also saw that there were no outliers. We similarly saw that scores that were grouped by percentile showed that some of the most extreme scores lied within the lower and upper percentiles, which proved another metric, provided another metric of reassurance that our PRS analysis was being executed as expected. And so a secondary assessment that we kind of applied to our results was to look at, does distribution vary across cases versus control cohorts? And so looking at a similar distribution but split by case versus control, we see that there is a significant difference between the case and control populations. And so we can say that public variants do explain some level of risk within the diverse cohort. And so another set of assessment that we kind of applied to our results was, does distribution vary across race or ancestry? And so looking at similar distributions, but this time looking only within individuals who self-identify as white, we see that the distributions kind of maintain a normality, and we see that in the case cohort that they're more further to the right, so there's higher PRS scores for these individuals, suggesting that public variance from groups with the same race or ancestry as the validation data perform well in our data. And so similarly, if we look within the proportion of our cohort that identified as Black, we see instead that if we look particularly at the red, the cases, that there's an uneven distribution of the PRS scores. And also that there is more representation of higher PRS scores, as can be seen here, versus like the lower. And so this suggests that the public variance from groups with a distinct race or ancestry versus as the validation data generates higher PRS scores. So they might be overestimating risk. And this might be due to just allele frequency and differences in like the population structure. And so one of the final questions that we had was, how much of a group's risk does sequence variation that is measured by PRS explain? And we can kind of look at the separation between the case and control, so how farly separated are they, to kind of answer this question. And so if we look at the effect size in standard deviation and like cases versus controls, we see that for the individuals who identify as Black, that there is less of a separation versus the individuals who identified as white. And so we can kind of assume that the distributions look a little bit more like this. And so we can interpret this to mean that the public variants explain more risk when the test and the validation cohorts have the same race or ancestry. And so for our next steps, we have been interested in integrating the environmental variables within our data. And so we're looking at, for example, area deprivation index, which is a available social determinant of health variable within our data. And we see that when we look across different populations, that there is a difference in distribution for the cases and controls. And so this suggests that area deprivation and zip code may be a social determinant of health with strong influence on race stratified risk. Okay, and so for our future directions, we would kind of be taking all of our taking notes from the initial analysis and wanting to repeat a lot of these analyses in our novel, more diverse cohort. So we want to generate our own base data, target data, and test data sets, and then validate within a comparably diverse cohort to see if we can explain more of the risk that's associated with sequence variation. Okay, so this is the last slide. We hopefully seek to ultimately generate a model that would fulfill the existing gaps within cancer risk modeling. So we would kind of want to fill in this gray area here. Thank you.
Any questions? I have one question while they're circulating in the audience. So how would you factor in multiracial groups?
Yeah, that's a great question. That would be kind of a situation where we would want to possibly use ancestry as a continuous variable and be able to kind of like look at like risk accounting for, like within our model, we would use like a continuous variable for that. Not continuous, yeah, continuous, sorry.
So if you're pulling it from, if you're pulling this from electronic medical records and, you know, how do you know the data is clean? Maybe some don't always check the box because my kids don't check the boxes and they're multiracial. You know, they check Asian even though they're Asian and white.
Yeah.
So.
That's a great question. I think that when dealing with like any type of like large data, you do assume that there's some amount of human error. But I think like if we look at things kind of more generally and like look at general trends, that could probably hopefully not be a like penetrating effect on like big picture conclusions. Yeah.
We'll take one question from the audience.
Hi, thank you for your talk. So I was wondering about, maybe I don't know enough about how polygenetic risk scores are made, but I was curious about your model of the case and control for the polygenetic risk scores in African American women. What did the bimodal distribution of the polygenetic risk scores suggest to you about the quality of using PRS as a model?
Yeah, I think we can draw like conclusions about using PRS in general, like after we've generated a new PRS like in a comparable background. So currently a lot of I think all of the published PRS variants were generated from GWASs that were entirely European. So if we're trying to use those variants to score our diverse data set, we're not sure if the distribution looking a certain way is due to just not having matched ancestry, or if it's just like a feature of the PRS analysis. So, yeah.
So how would you distinguish your results in the end? Like if you take into account this new data set that you're looking at, how would you attribute the results to either being that the PRS scores themselves are not indicative of the group that you're studying, or that something is not accounted for in your data set?
Yeah, I think that after generating like a new PRS in a diverse cohort, if we see like persistent, like over, or like it's not explaining all of the risk, then at that point, we are interested in including social determinants of health. We're including other covariates that tend to vary with race and ancestry. And so that would hopefully provide some insight. But yeah, I think with you know, when doing science, like there's always like questions and like, is this explaining everything? So we're hoping to have answers to that question, but we're open to like still having more questions after all the data is collected.
We have to close out, just flash talk, so maybe afterwards. Yeah, sorry about that. We'll have a closing comments, maybe we can have more questions there. Sorry about that. Thank you so much for your talk.
Thank you.
Okay, next up, we have a change in what's in the program. So if we're going to welcome Dr. Imad Karam. I hope I said that right. Dr. Karam is a Chief Hematology Oncology Fellow at SUNY Downstate, right here. And currently he's completing his fellowship training and is an awardee of the American Society of Hematological Medical Educators Institute. So he's got a wide range of interests, including breast cancer, medical education, cellular therapies, and advancing equitable cancer care. Thank you.
Hello, everyone. Thank you so much for having me today. As you mentioned, for full credit, I'm presenting on behalf of Dr. Bashan Hamadi, who unfortunately tragically lost her father this week. So I did work on the study, but she did most of the heavy lifting, and she is our research leader. So this study is entitled Impact of Diabetes and Glycemic Control on Pathologic Complete Response in Breast Cancer. And it is a retrospective cohort study in Afro-Caribbean population, which is the majority of the patients that we actually see in our clinic. So there are trends toward lower pathologic complete response in patients who have diabetic patients, and also specifically those who have uncontrolled diabetes as well. So the question is, does diabetes or baseline HbA1c impact pathologic complete response after neoadjuvant chemotherapy? And we had clear numerical trends to show that lower PCR in diabetic patients in 52 versus 75%, and the higher baseline A1C in non-responders, 8.05% versus 7.19%. Although the results did not reach statistical significance, however, this has been limited by the small sample size. But the implication of this study is really big, and the directional data aligns with preclinical mechanisms of chemoresistance, warranting larger, adequately powered multicenter trials. So, in general, we give neoadjuvant chemotherapy in breast cancer population who have a triple negative or HER2 positive disease more commonly than ER positive alone. And almost universally, all patients will receive neoadjuvant chemotherapy. A pathologic complete response means after doing the surgery, on pathology, we don't find the cancer, or the cancer has responded to the chemotherapy. So there's no residual cancer. And this factor is a very important factor as a prognostic implication on the impact of second line of treatment and long-term survival, as well as event-free survival, which means recurrence. So the biological hypothesis behind that is that hyperglycemia does fuel chemoresistance. And diabetes in breast cancer patients is associated with worse cancer-specific survival. Overall survival rates are lower for diabetic cohorts, and diabetic patients experience increased treatment toxicity and altered treatment patterns. And the mechanism behind that is that hyperglycemia does alter the AKT and mTOR signaling pathway and increase anti-apoptotic signaling, which means the cancer cells can survive longer, their proliferation is enhanced, and their potential to invade is also increased. And this incur chemoresistance and reduce the effect of neoadjuvant chemotherapy. There is a disproportionate burden in Afro-Caribbean populations with the high prevalence of metabolic comorbidities, including diabetes and poor glycemic control, and the higher breast cancer mortality rates. Now, the intersection of these two risk factors makes understanding the relationship between metabolic health and PCR urgently necessary for risk stratification and treatment optimization in Black and Caribbean women. So this is a retrospective cohort selection, and we had 153 patients who received neoadjuvant chemotherapy with documented chemotherapy regimen. 52 patients were analyzed, and they had complete charts without missing data for PCR and diabetes status. Our primary outcome was whether those patients achieved PCR or not, and our secondary endpoint included diabetes status with defined by medical records or A1C above 6.5, and also a baseline A1C at diagnosis and its effect on the response. Non-diabetic patient exhibited a 22.5 higher PCR rate, although it's statistically significant, but the trend is obvious. With 75% or 12 patients had PCR versus 52% in the diabetic cohort or 40 patients. In addition, patients who had a lower response rate had the higher baseline A1C levels, with most patients had a baseline of 8.05% in 11 patients without PCR versus 7.19% of, which is 26 patients of the whole sample. And the cohort exhibited a broad distribution of glycemic control, with many patients presenting above the 7% threshold for good control. Now, diabetes was not an independent predictor for PCR after adjustment. However, it does show a trend favoring worse PCR or worse outcome with patients who have diabetes, as well as patients who had higher HbA1c. Some of the advantages of our study include utilized real-world clinical data from a defined neoadjuvant chemotherapy cohort, included both binary disease status and specific glycemic control, and applied rigorous classical statistical methods. Obviously, the limits include a small sample size, severely limits power to detect modest association, and this study is done in a single center retrospective design, which can introduce potential confounding factors. And there, the charts are not sometimes written in a way tailored to do those retrospective studies. So we had some missing data, including the regimen, the adherence, and specific tumor subtype. For future implications, definitely we would like to have a larger data set, including multi-center cohorts with complete metabolic data to achieve necessary statistical power, probably a deeper understanding of the biology, time-varying metrics, including tracking the glycemic control continuously during the neoadjuvant chemotherapy period, rather than relying solely on the baseline HbA1c. And on the long run, hopefully, we'll have interventional trials with prospective studies that target patients who have diabetes with aggressive, multidisciplinary teams to control uncontrolled glycemia and potentially achieve better pathologic complete response and subsequently overall survival. Thank you.
Any questions from the audience? Yeah? All right.
I just want to ask, again, my name is Hara Prasad. Thanks for a great talk. You're presenting on behalf of someone, so you both have worked. So the question is I found the age is you have given a very short, this one, but age-related diabetes mellitus is also significant, right? Because the more you get age, the more you get HbA1c to be higher, right?
I don't think it's the age is related to.
Yeah,
this is a very good question. I don't think it's.
There is always in the articles, journals you read age-related diabetes mellitus mostly get that. So I found the bar is a little bit smaller.
Mm-hmm. Yeah.
So.
I mean, this is a very good observation. Again, this is limited by the sample size. That's why you're seeing a smaller bar. But again, the focus here is on the diabetes more than the age. Obviously, with older people, age will factor in, especially if they have other comorbidities and their tolerance to the treatment. So this probably is the confounding factor here. Good question.
Hi. Quick question. Maybe I missed it. Did you classify the subtypes, the breast cancer subtypes?
No, in this study, they were not classified. Okay. This would be like a future study that we would like to work on, definitely.
Yeah, just a short question. I don't know much about breast cancer, but I was just wondering why most of the patients, I think it was about 80% of patients have diabetes, right? So is there any correlation between the breast cancer and diabetes? If so, I think maybe there's some difference in the breast cancer stages with diabetes. So did you do any analysis?
This is a very good observation. The study did not really stratify or did not have a subgroup analysis. The question was just simple to assess whether diabetes is affecting the PCR, but definitely it's something that we could look into for potentially future studies to compare subtypes and if any relationship with advanced stages in patients who have diabetes. Thank you.
Thank you for your talk.
All right. Thanks.
Thanks so much. All right. Last but not least, we have Sun Kim. So Sun is a PhD candidate from the Tuvison Laboratory at Cold Spring Harbor Laboratory. Her research focuses on pancreatic cancer biology, particularly KRAS interacting proteins and mechanisms of therapeutic resistance. So today she will present her work that investigates resistance to RAS inhibition in PDAC. Thanks.
Hi, everyone. So last talk, I'll try to make it quick. So I am a PhD candidate in the Tuvison Lab, and the work I'm going to show today is going to be a little bit of a pivot from the work you've been seeing probably recently in the recent talks. We are a pancreatic cancer lab, and we focus on multiple mechanisms, either biomarkers, tumor microenvironment, in my case, RAS inhibition. So I'll be showing you a vignette of the work we've been working on. So we're a preclinical model stage. So how the workflow normally is we do all the preclinical work, publish it, and then this could set up kind of a framework for future clinical trials. So today I'll be sharing with you some of the work we've been doing with our RAS inhibitors, RAS inhibition in murine models. So pancreatic cancer, actually, if you see here, it has one of the lowest 5-year survival rates of almost 13%. And actually, when you compare it to 1995 to 1997, it was 4%. So we've come a long way, but there remains work to be, improved. And particularly, one of the reasons why pancreatic cancer has one of the lowest survival rates is the lack of detection. By the time it's diagnosed, I'm sure many of you guys may have someone who had pancreatic cancer already. It probably had already metastasized to liver, lung, and by then there's no therapeutic really efficacy on any of this, and only palliative care. And the most important thing is the current first line of therapy for pancreatic cancer is chemotherapy. So this is important to note that there's no targeted therapy. And targeted therapy is usually important because you're targeting the driver mutation as opposed to just, chemotherapy kind of targets both your normal and malignant cells. And pancreatic cancer is actually really unique compared to other types of cancers where a majority of the patients possess a driver mutation in KRAS. And KRAS is a unique mutation where a lot of the patients, compared to colorectal cancer or lung adenocarcinoma, you can see that a lot of the KRAS mutations are being driven either 43% by the G12D. So this means that the G is mutated to a D, and then 32% is being driven by the V mutation where the G is mutated to a V. So obviously this addresses a concern for, is there a way we can toggle this and see if A, is there a response, and B, are there resistance mechanisms that are associated with this? So in our lab, we have 2 models. I'm not gonna go into the nitty-gritty details, but one is a KRAS G12V model and one is a G12D model. And as I mentioned before, D and V are the top 2 mutations that are found in pancreatic cancer. And the first part I'm gonna talk about is the G12D, the KVPC model, which is a murine genetic modified model that we have in our lab. And you can see that this is a response curve of the tumor volumes and survival curve. So you can see that around that 15 days, the tumors do get smaller, but you can see that those steep curves at the end, the growth, it indicates a sharp growth, which A, indicates they're no longer responsive to the G12V deletion, and the cells have found a way to become malignant and overcome this. And obviously, although there is a survival advantage, most of them eventually succumb to death. And obviously, one of the more important causes or indications is we were interested if at endpoint, if there's any mechanisms that we can potentially leverage with this RAS inhibitor. And this is just to show you that the mutant allele is deleted. So even though they only have the wild type RAS, wild type KRAS you guys all have. So obviously, despite it being deleted, these mice are succumbing to death. And just this could be busy, but I'll try to take it little by little. So the left side is just showing you that we deleted mutant KRAS. So there's no mutant KRAS and only the wild type RAS is present. And if you see here, this is we can do stainings of certain proteins on the mouse tissue, tumor tissue. So what you can appreciate is it's mainly green here. So these are staining for different differentiation markers of the tissue. And you can see it's more green here, but at endpoint, when they relapse, they're a bit more yellow, where a combination of red and green results in a kind of a yellow hue. But more importantly, I think the point you can drastically see here is these white dots and the redness. So red stands for something called phosphor-ERK. So it's kind of an activation of RAS signaling that's downstream. So you can see more phosphor-ERK and just more malignancy in these relapse cells. So we were interested if what the program was. And what you can appreciate is these are called Western blots, where you're doing a protein assessment of these tumor lysates. And you can see that even though we don't have any mutant KRAS, you can see that at the end, at the relapse time points, they're elevated. So this means that even though there's no mutant KRAS left, the wild type KRAS protein is getting increased, and the same goes for the HRAS as well. And you can kind of see there's a mild increase in the phospho-ERK, which means basically, even though you mutate or target the mutant allele, there's some oncogenic reprogramming that's happening. And I think one point with the KVPC model that I just showed, it means the resistant endpoint have increases in wild-type RAS. So we're curious about, the auxin, the G12D model, is it the same thing? And it's a busy slide, I apologize, but basically what we're doing is showing the same thing where this is a cell model, so we have to transplant these, and we treat them at a certain enrolled size, and you can see that even when they're deleted, similar to the previous model, you do see a certain advantage when you do delete the mutant KRAS. Tumors respond nicely, so the yellow going down means that the tumors shrank. These are just waterfall plots that you just show to show best response of the tumor. But obviously, what this is doing is these curves, the green is just going up, so that's the control. So the tumors are just growing, they don't care. But when you delete, which is in the orange, you can see a steep mountain kind of like going down, and then a lot of them, the velocity is going up, which indicates that the tumors are coming back despite having no mutant KRAS, which is interesting, right? Because in the beginning, I mentioned that mutant KRAS is one of the main driver mutations of PDAC, but we're deleting this, but it seems like they're reprogramming themselves to become more oncogenic and escaping. And this is also a staining of the tissue, the tumor tissue, for example. And on the left is vehicle, so it means they didn't get any deletion. The right is where we deleted the driver, the bad allele. And what you can appreciate is basically at endpoint, you can't really tell differences between the vehicle and endpoint, which signifies that despite a certain survival advantage, the tumors look similar to vehicle. And obviously, we were curious if this happened earlier, and we did something that was more acute, shorter, to see if there was a response. And what you can appreciate is, at least up until 7 days, the phosphor, which is an activation of the KRAS signaling, you can see that the orange, purple, and blue dots are down, which means that the drug's working. Like the cells suffer because there's no KRAS. And the Chi67, which is a proxy for cancer cell proliferation, is also down. But what you need to appreciate is at endpoint, which I showed earlier, they did not care. There was no difference between the vehicle and endpoints. So overall, it seems like these shells are shifting where early they're responding, and the Chi, which is the cancer cell proliferation, and the output of KRAS, signaling seems to be down, but then at endpoint, which is in orange, they seem to be kind of relapsing and going up. And obviously, this is also another Western blot, where we can check for protein of certain lysates or cells. And what you can appreciate is, despite having no mutant KRAS, so made means what I deleted, and endogenous signifies the wild-type RAS allele. And what you can appreciate here is the KRAS on the left. You can see darker bands, right? So that means that there's a higher expression of the protein. And same goes for the last three rows for the HRAS and NRAS, which signifies that despite having no mutant KRAS, you have wild-type RAS isoforms basically going up, which is really ironic but cool in terms of trying to understand relapse. And the right side is just a different way to depict this. So the left is protein and the right is RNA. And basically they show the same direction. So obviously, I think a converging point in both of these models is that resistance at endpoint in both models results in upregulation in wild-type RASes, which is really important. However, there's already been a lot of clinical trials targeting monoallelic KRAS, and kind of the work that's been coming out, especially with the lung adenocarcinoma patients, is the monotherapy, it works. It's going to move the needle, but it's not durable. And I think the work that we're doing all the more shows that you need other targets in combination to potentially prolong the relapse and increase the survival. So in summary, I think it's important to really just not think about monoallelic therapy. PDAC is really unique in the sense that a majority of 90% of patients have this mutation, but despite targeting the mutation, so taking off the leg, basically, they still learn to survive. So we need more. I think this is going to be an incredible field that's been really ramping up within not just the PDAC field, but the RAS field in general. And now we have pan-RAS inhibitors, which means pan means it targets all the RAS. And even with that data, I think that's coming out in the clinical trials it's not. The relapse is inevitable. So I think in the long run, I think we're going to have to leverage it with other combination therapies. So that's all, and I'll take any questions. Thank you.
So impressed how this group all stuck to time. All right, do we have a question from the audience?
Hi, thank you for your talk. I was curious, because you seem to show that the total, there's this compensatory effect of wild-type KRAS elevating after the deletion of the mutation. Have you considered this is more of a thought experiment, have you considered a mechanism where the cell is actually trying to restore the previous levels of RAS that it was in the cell before the deletion of the mutant KRAS? So then have you considered maybe replacing the protein RAS with some sort of maybe inactive version of the protein to trick the cell into thinking that there is a sufficient amount of RAS in it after the deletion of the mutant?
Mm-hmm. Yeah, so that kind of delves into or touches upon the kind of compounds that are coming out. So I showed you a genetic deletion model where if you delete it, like you said, the phospho-ERK, which is that proxy of RAS signaling, decreases, and obviously we do see eventual rewiring with the RAS. But I think what you're asking in your latter part of your question is what happens if you, instead of delete or degrade it, what happens if you just block it, for example? And I think one point that I want to make is there's a lot of the compounds that are coming out with these RAS inhibitors, there are degraders. There are small molecule inhibitors. For example, the Revolution Medicine, they're bound to a CYPA, which is like the payload, where they just block it. So the protein's still there. They just can't interact. So overall, I think despite whether it being a degradation, inhibition, or whatever, all converge to kind of this upregulation or rewiring where the RAS proteins do increase. So I think there are some clinical trials that are getting started right now with wild-type RAS inhibitors, with the pan-RAS, with the monoallelic together, and actually suppressing it together actually shows an even better survival advantage than either monotherapy. So yeah, that'd be interesting.
Thank you.
Right here, down here.
Hello, my name is Hara Prasad. Great talk. Eventually, your name is also similar to SUNY Downstate. Yeah. Yes, he's from where? Okay. So the question is recently somebody came from your institute, it's like Cold Spring Harbor, where because most of the people who work on cancer, and you have a good mass spectra instrumentation in your institute. Mm-hmm. So I think that as you said right now, that in spite of inhibiting completely, not deleting, partial inhibition you can do, and then you can do a mass spec on that, because all the libraries are there, and you have to see like which other proteins are uplifted, which was down, now it is uplifted. So by that way, because survival is like not only you are trying to inhibit some other targets, but you have to see that what other genes are coming up when you're inhibiting that. So that can be a good approach.
Yeah. So I guess just to rephrase your question, you're asking, if you do a partial inhibition instead of complete deletion of this protein to see upregulatory pathways. Yeah, one of my second projects do delve into this. We do see a rewiring of like the protein interactome. Unfortunately, at least from the experiments we've did, because KRAS is such a potent driver, deleting those could potentially not be as helpful unless you combine it. But I think one thing I didn't really talk about is pancreatic cancer is not just cancer cells. It has a whole tumor microenvironment with fibroblasts, macrophages, immune cells. Those all play a role. So what I'm showing you is just literally one tiny part of what's going on in the tumor. I think overall, everything's going to be both cancer cell intrinsic and extrinsic. Yes.
Thanks. Great talk. I was wondering, is human studies show that there's sex-specific differences in pancreatic treatment? And I'm sorry if I missed it. Are they male mice, female mice? Are you looking at sex differences?
Yeah, absolutely. So the first part of it is both male and female. So we just look at it separately, and rarely are there differences, and then we combine it. I will say, because the first model is driven by a tamoxifen model where we can toggle the mutant, women have more estrogen receptors, so obviously that could potentially play a role in the tumor microenvironment, but it seems like there's not huge differences because the amount of tamoxifen we dose isn't that much. And I think in terms of your question to known mutations that are more specific to like male or female, I don't think so, but it does seem like the men tend to have worse prognosis. So yeah.
All right, thank you. Thank you for a fabulous talk. Thank you everyone for your questions.
All right, I think we've come to the conclusion of all these sessions and the day for us. All of you did great. You were able to sit through all these lectures till 3:30, and we were able to do well with time. We started quite late, but we made it on time, so that's very good. But first, I would like to thank one person in the audience who has made everything possible. Dr. Laura Martello Rooney. Could you stand up? Okay, and my partner at Cold Spring Harbor, Dr. David Stuyvesant. He's not here today, but he gave opening remarks, and I believe you all saw him. I would like to thank him for his support for the program. Obviously, Northwell Health. Thank you again for coming, all the participants from Northwell Health. And most importantly, to all the participants in this audience, this is such an important discussion around what we need to do to understand these biologic differences. That's the main thing. We want to be able to understand what are these differences in biologic differences that we can actually focus on and then make a difference in health disparities, cancer health disparities. And then hopefully we can also get people together once in a while every year to create these connections. And I think these connections will end up with something. And that something always ends up with, some work will be done later on to address questions that are raised in this conference, symposium. And hopefully we can follow through with those questions, write NIH applications, get them answered, and come back and present to our colleagues and also to the community. So having said that, we will be having the 12th annual Cancer Health Disparities Symposium next year. And then we'll be inviting you back, and I hope you would accept the invitations. And for those of you who are here, please spread the word. We want everyone to know that SUNY Downstate actually hosts this symposium every year. It's a very important symposium. We want everybody to know, and then we want you to spread the word as much as you can. So with that, I would say have a good day. For those of you who have come from far away, have a safe trip, and I will see you next year.